right, yeah. So I'm going to just give you a brief. Initially, I thought I had 15, but now I have 10, so I'll rush it. Yeah, so to just share, what's going on at UVRI in the space of, emerging, biotechnology. And I've got this information from the IBC, which I am a member, but I'm also an investigator, and I'm working on 2 projects that I'll share because they're captured here. Yeah. So first of all, the Uganda Virus Research Institute, its remit is on, infectious diseases. That's what they focus on. But in infectious diseases, they focus on the pathogens or the parasites that cause the diseases, but they also focus on the vectors or the insects that transmit those diseases. So that's where our research is targeted to. And of course, the aim of the research is to increase our understanding and our knowledge so that we can be able to develop better tools to control them better. As you all know, vector-borne diseases, they're still a problem in our part of the region, and we need to be doing more to control them. So that's where the research that we do at UVRI comes in. So when it comes to the actual pathogens that we focus on, of course, viral pathogens are a big part. It's part of our name, but we also look at other things, beyond viruses. So we look at parasites, we have worms. We also have parasites that cause, other diseases like malaria. And then when it comes to vectors, we mostly focus on the mosquitoes because they are some of the most notorious, disease vectors that transmit a lot of things, arboviruses and also malaria as well. But we also focus on some ticks as well because they also transmit, a number of pathogens that affect people and human. And some of the diseases that we are looking at, like things like yellow fever, malaria, CCHF, that Crimean Congo hemorrhagic fevers, and others. And we do a lot of work with vaccines as well as another approach for control. And all of those angles require biotechnology and therefore IBC oversight. So we have a long history when it comes to biosafety at UVRI. As early as the 60s, we were handling, CCHF, and then our first biosafety committee was formed in around 1997. But before that, there was an actual safety committee, but the one that's aligned to, the listed regulations. We can date it back to 1997, and they have oversight on the lab work, waste disposal, chemical handling, radiation handling, and all those things that are part of the research. But we also have an actual institutional biosafety committee that was registered in 2021. So that's the membership, and those are some of the people on the committee. They bring, diverse expertise for an institution of our kind. There you have molecular biologists, you have virologists, you have immunologists, you have geneticists, and lots of people with, other background that's needed. So now when it comes to the emerging biotechnology, I've picked out a few projects just to share on what's happening. So one of the projects, the one that's probably most advanced, I think, that we're having is the one for target malaria. So in target malaria, they're seeking to modify, the malaria transmitting mosquitoes to reduce its numbers. So the focus here is to modify, to, of course, reduce their ability to reproduce through different strategies. But the whole underlying idea to that is if you can sort of interfere with the reproduction and you curb the numbers, you target genes that are essential in the reproduction or even the development, then that way you can reduce the numbers of the mosquitoes that are doing the transmission, and that way you can curb the disease transmission. So that's the biggest idea for target malaria. And they're trying to use some innovation when you're doing that. So they're making it a tool that's going to be self-sustaining because it's going to harness gene drive, and gene drive is a mechanism that sort of preferentially selects for the inheritance of particular traits of interest. We talked about traits this morning. So here it will be a trait that targets, either disrupts those genes essential for fertility or developing into a certain sex or the other. And at the moment, that work is phased and it goes through different stages. Of course, we start with technology strains that are of lower genetic complexity before we go to the final ones that have actually drive, because that's part of the development strategy that we pursue. And at the moment, I would say for the work at UVRI, we are at the stage where we are handling a non-driving strain, but modified still to target, development. And we are looking at it at the lab stage. So that's the stage we call containment. That's the stage before, you go to the next level, which is, field evaluation, which will involve actual releases. So we're still a step back. And so we have all the infrastructure set and all the people trained and all the protocols developed and all that kind of thing. Then there's another work that's still in the project formulation step. So that's still genetic modification for mosquitoes, but this is using different technology. And here we're looking at ability to modify so that, we can still affect the development of the mosquito, but this is targeted to just the females. So there we modify and we put what you call repressible, mortality for the females. So they develop, and at some stage they cannot complete the development of their phase when they are in the field. But in the lab, there's a way we can, repress that so that it's not active. But outside, they will not have that. So that's when it will kick in. But this is still in the early stages. We're still formulating it. But that's work also that we'll be seeing that will be happening. Then we also have work on snails, and this is exotic because some of the strain that we plan to use, are not local, but the whole idea is we still need to contain them, and of course, once they are established, then we're looking at behaviors that will involve, infections with worms and things like that for them. Then we have a lot of vaccine work where we are using recombinant technology to look at different vaccine types. So we have various COVID vaccine where we're looking at those that are vector. Luckily, the one who spoke in the morning introduced to you the vehicle, so that's what they call the vector. So you have a construct that has the vector as the vehicle, but then in there you put an insert or a gene of your interest. So here it would be messenger RNA that will elicit resistance probably to the COVID. And then we also have others which use just the routine monoclonal, and they're part of construct that we're looking at to develop these kind of vaccines for COVID. And then we also have vaccine work that's going on towards the tick. That's also happening. And then we have other vaccines targeting Rift Valley fever, Crimean Congo hemorrhagic fever, and even HIV. So all that work package, of course, uses biotechnology, and of course, it's under the oversight of the IBC. And then there are other work that we do, whereby we look at, we manipulate different things, and we produce infectious things and diagnostic things. So all that is also part of biotechnology, and that's taking place at the Institute. So a session by some of the members of the IBC, and I think with that, hope I've not gone over.